From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on January 25th, 2024. Hello, Micronauts, and welcome once again to today's Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today, believe it or not, is the 26th episode of Matters Microbial. As always, I am grateful for every listener, viewer, and commenter. You make all of this possible. First, an exciting new member of my Wunderkammer. When I podcast from my office here on campus, I tape a sign to my door. It's very high tech. I thought about trying to create some kind of lighted sign, but I am not an engineer. So imagine my surprise when my highly creative mathematician wife created a sign for me to put on my door. It's lit with colored lights and a switch. And naturally, knowing me, it glows in the dark. It was quite a surprise, and I am a very lucky microbiologist. The first full week of my introductory cell and molecular biology class was a lot of fun. It is a large class by our standards of about 48, and you can see it here. I try to find things that will engage and amaze them, kind of a microbial shock and awe. Our cure-based lab, searching for antibiotic-producing microbes, requires these first-year students to learn basic microbiological techniques. So I use a very bright strain of the marine bacterium, Photobacterium. This culture flask glows very brightly. I even encourage the students to pose with the glowing flask to create a Lux selfie. But I use these luminous microbes to show students how to streak a plate to single colonies, a basic technique. Now first, this Photobacterium strain grows very quickly, so the students can evaluate their skills promptly. Plus, it glows in the dark. The students are working hard and learning a great deal. Something that I try to teach my own microbiology students is how few microbes in nature can grow on standard nutrient medium. Turns out that microbes are very finely tuned, both biochemically and genetically, to even very small differences between environments. And some microbes prefer very low nutrient conditions, which is termed oligotrophy. This was described decades ago as the Great Plating Anomaly. I'll put a link in the show notes to this classic set of observations. Microbiologists continue to fine-tune the ecophysiology of microbes to laboratory conditions, but it's an ongoing process. And it's a far cry indeed from simply plating things on nutrient plates. After all, nature is not a culture tube nor a petri dish. Despite what I thought growing up, the ocean is not necessarily rich in nutrients. Thus, it's a challenge to grow and study all of the rich microbial diversity to be found in marine environments. Here is the name of an organism every micronaut should know, Pelagibacterium. And you will get to hear all about this organism today from our guest. Dr. Cameron Thrash of the University of Southern California's Department of Biological Sciences in Los Angeles is an expert on the kinds of microbes that live in marine environments. It's such a treat to hear from him today. Welcome to our Quality Quorum, Cam. It's truly an honor.
Thank you, Mark. Nice to be here.
I am really looking forward to this because actually the history of microbiology was originally marine microbiology when they first started looking in the environment. And Southern California at Scripps Institution of Oceanography and Zobel and all the rest, it's always been a special interest of mine. So I guess a good thing to tell people as we start is what brought you to your current position, just so they have some idea of how you got here.
Yeah, so I started off as a college student at UC San Diego thinking that I wanted to be a scientist, but I had no idea what that meant. And along the way, I actually got enamored with a number of other things that sort of took me off that path. And I wound up after college unemployed, but with a biology degree. And needed to get something in line that would give me some health insurance and stable income. So I applied for all the positions to which I was qualified, which were few since I had no lab experience other than the canned labs that we had to take for class. And I was hired by Dr. Monica Carson at the Scripps Research Institute, which is not the same thing as the Scripps Institution of Oceanography, but they're very close. The TSRI is just up the street from SIO. And I got my first job as a entry-level research technician. And starting that job, I absolutely fell in love with academic science and basic research and the lifestyle of being a scientist. And I realized at that point that that's what I wanted to do for my career. And so at that stage, it was sort of a matter of what would I want to specialize in, and how would I make that happen? At the time, I was doing neuroimmunology, and I realized during that work that I wasn't really interested in chopping up rats and mice and doing all the transgenics and all that. In other words, I wasn't super interested in biomedical research. I have always had a love for the ocean. I've always had a love for sort of being out in nature. And so I wanted my work to be focused on environmental research. And I discovered a field at that time called bioremediation, which is the cleanup of contaminants using microbial metabolism. And I was totally enamored with this. And I decided to try to find ways to become to do a PhD in a field that would link up with bioremediation. And so that's what led me to grad school. In grad school, I did work on bioremediation in the lab of Dr. John Coates at Berkeley. And in that time, I learned a lot about microbes. I'd never taken a microbiology course before I started a PhD in microbiology, but it all sort of got absorbed very quickly, and I studied a lot because I was passionate about learning. And as I learned more about the world of microbiology, the more I got just so much more excited about understanding microbial evolution, microbial ecology, all the various aspects of microbiology that are out there. And so I chose to do a postdoc in more of a basic research as opposed to applied research like bioremediation, and start studying the organism you already mentioned, Pelagibacter, in Steve Giovannoni's lab at Oregon State as a postdoc. And at the time, I also was learning bioinformatics instead of just doing my PhD was all physiology. And I think somewhere early on in the pursuit of the PhD, I really thought that being a professor was going to be the goal. And so my through the career path there, my intention was to pursue an academic post. And so those are the types of jobs that I applied for during my postdoc, and I got my first position as a faculty member at Louisiana State University in 2013. And a few years into that, I was lucky enough to have the opportunity to move my lab to University of Southern California, and that's the hopefully Cliff's Notes version of how I got to be where I am now.
I think it's important for viewers to know a little bit about that process. I mean, there are lots of different routes. And one of my fears is that people will hear about just one route into science. And I guess the take home that I try to present on the podcast is just as there are different faces and different voices, there are certainly different paths. So thank you for sharing yours.
Sure.
I did want to point something out that makes you absolutely essential to the scientific community. And it's a 100% true. I believe that, Cam, you get up at about 2 a.m. and you start scanning for papers, because every day on social media, there are links that you present to all kinds of papers. And may I point out, not simply in marine microbiology. You have very broad interests, and it shows. I cannot tell you the number of times that you have presented me with a paper that informs some of the work that I do or what I teach. So I think a lot of people should follow you to learn more about what you see. You're a curator.
Yeah, uh, you know, I don't get up at 2 in the morning. I get up about 5, but not 2. And I appreciate that you find value in this. I think this all started off in a very selfish way when I was a postdoc because I was paranoid about missing the literature. I was paranoid about being in a new field and not being up on topics. And so I joined social media, and I also started scanning table of contents really early on. And over time, it sort of built into a way for me to help promote our lab's research, which so there is some definite selfish interest here. The more followers you have, the easier it is to promote our research, but that helps my trainees, that helps the people that work in the lab, that helps everybody that's associated with us. And um, yeah, I think at this point it's a community service to some degree, and I'm scanning the literature anyway, so there's no reason not to push it out there at the same time. So it's really a I don't know, it's been a great way to sort of interact with the community, and I found that social media has been a really wonderful uh, augmentation to the scientific enterprise for forming collaborations, meeting new people that have shared interests, and uh, being able to generate new possibilities for our research. So it's definitely a two-way street.
Well, I'm absolutely going to put links in the show notes to your social media contacts so that if anyone's interested, I promise anyone viewing or listening, you will learn a lot if you see the kinds of things that Dr. Thrash puts out there. So I guess what I would be helpful if you would just give, and I hate this term thumbnail sketch because I have no idea where it comes from, but the kinds of work that you do. I love your website. Again, I'll put a link to that in the show notes, where you're basically like searching for microbes in the ocean environment.
Yeah, I think that's not a bad way to put it. We definitely don't have one specialty or one focus. We have multiple things going on. And some of that is a legacy of being a new assistant professor many years ago now, and just trying to get funding in as many different avenues as I could. And some of those actually wound up sticking, and we've been pursuing them ever since. Um, but it's also partially because it's one of the fun things about being a faculty member and running your own lab is the ability to sort of choose the types of things you want to research and investigate. And I've been able to be, yeah, I don't know, I'm curious about a lot of things, so we go in different directions. So I think the very general idea is that we are interested in understanding the function of microorganisms in aquatic environments primarily, although I have worked in other environments. And our primary foci right now revolve around sort of a subset of topics. So we have some very lab-based research on individual microorganisms or individual taxa, learning more about their cell biology or their physiology or their genomics. One of which we have a major focus on is SAR11, and I think we'll talk about that in a little bit. But we also have some sort of system-based research where we're interested in understanding how the system is working or how the microbes in the system is working. For example, we have funding to study coastal hypoxia, areas where ocean water is depleted of oxygen, and the microbes are the ones that do that oxygen depletion. And while we may have a good understanding of sort of some of the sort of large scale causes like eutrophication, nutrient influx into coastal areas, stratification, physical processes, there's very little known actually about which microbes do the oxygen removal. And therefore, we have a lot to learn to gain predictive capability in areas where oxygen is low in the oceans. Because again, yeah, the microbes are removing the oxygen. So they're the business end, if you will, of the process. So we're studying that in multiple locations. We've spent a lot of time in the northern Gulf of Mexico, and we've had a series of cruises there that concluded this year. We're starting new cruises in the Southern California Bight starting next year, or actually this year, sorry. We're in 2024 now. And so those are some of the systems that we're exploring for hypoxia. We're also involved in a collaborative grant with Eric Webb's lab here at USC and Dr. Naomi Levine, Liz Kujawinski, and Max Sato at Woods Hole. And the five of us are investigating the microbiome of trichodesmium, which is a cyanobacterial nitrogen fixer that's very predominant in surface oceans. So my lab's role is to try to sort of dissect the heterotrophic community that floats around with trichodesmium and start to understand the interactions between these taxa. So those are some examples of things that we're doing that are not single organism centric, that are a little bit different than our focus on individual organisms.
I have to tell you, I'm really pleased to hear about the last project with tricho, because it's a fascinating organism. It looks beautiful, but what's interesting is that many things in the ocean are just surfaces. And so you're naturally going to have other organisms colonize those surfaces. And we're very reductionist in the way that we look at things, and it's been productive, but after a certain point, you have to think about the interactions. I have deep concerns about large organisms in particular and the assemblies that attach to them everywhere. And this is, I'm glad you're looking at it. Not a lot of people are looking at that kind of thing.
Yeah, I think, well, it increases the complexity of the research by orders of magnitude when you have to start thinking about not only one, but several organisms and how they're exchanging small molecules and how do you track all those things. So I personally see or perceive that there's a much greater emphasis on interactions, especially in systems that can be broken down into component parts or potentially subcultured out and then have those subcomponent organisms recombined in synthetic ecology settings. That seems to be a growing area of interest, and there's a lot more papers and research coming out in that realm. So it's an exciting time for sure to start increasing the complexity of the analyses and following that all around the world.
It always makes me laugh, and we've had this conversation at conferences when people started to study biofilms. They would look at one organism on a surface, and I would reasonably point out that's not really true in nature. And they would say, okay, I'm looking at 2 or 3, and then I would just give up and leave the room. But you have to start somewhere. And as you say, it's orders of magnitude more complicated, but it's the exciting part of it. I mean, as you say, it's a wonderful time to be a microbial scientist. Really is.
Definitely. Yeah.
So I'd like to, because I think a lot of things are going to focus on this organism that I promised to discuss. And the first thing that I would like you to explain is the difference between SAR11 and this term Pelagibacter that we've thrown around.
Sure. Well, SAR11 is the original name for this group of organisms. And the term comes from a clone library sequence that Steve Giovannoni discovered back in the very early 90s. And SAR comes from Sargasso Sea, where the sample was collected in the northern Atlantic. And 11 was just the 11th clone library sequence. And so there's actually a lot of SARs out there: SAR 324, SAR 406, SAR 116, SAR 11, SAR 92. They're all very different organisms, and some of those are separated by vast evolutionary distances. So don't get them confused. The SARs don't imply any sort of phylogenetic relatedness. They just all come from this initial survey or subsequent ones. So that's where the original term comes from. Now, during my postdoc, we were involved in analyzing a number of genomes of SAR11 and doing the first pangenomic survey of SAR11. And we realized with that research that SAR11 actually constitutes probably something like a taxonomic order, which we proposed as the name Pelagibacter alales. Now, the reason why we use that term was because the very first isolated SAR11, a strain called HTCC 1062, which was cultured by Microbe in 2002 in Steve Giovannoni's lab. The name for that they proposed as Pelagibacter ubique. And so that's sort of still a candidatus name officially. But because that was the original name of the strain, the first strain that was cultured, we've kind of kept that for the term for the rest of the clade. But it's important to know that Pelagibacter is just one genus, and there's probably dozens or more genera in the Pelagibacter's order, although most of that formal taxonomy has not been done. Some of the groups that we've studied in my lab, we've tried to propose sort of genus names or similar, but the way taxonomy is going right now, who knows if any of those will stick. But anyway, so that's the difference between Pelagibacter and SAR11. Pelagibacter is a genus within SAR11, and it also is the OG SAR11 culture, and so it's the best known.
So because of this, that species name ubique, based on, I'm presuming, the term ubiquitous, because you find it everywhere.
All over the place. Yeah. Yes, that's definitely where it came from. Yeah.
So my question is, about this organism. It's extremely small. It's extremely fine-tuned to low nutrient conditions. You heard me speak a little bit about oligotrophy. By the way, that's something that students always have a little bit of difficulty with when I describe it. This idea that some organisms don't like a big meal. Maybe that's the way to think of it. But I guess a better way is to say that these organisms have been fine-tuned to what's necessary to survive under those conditions. Would that be a good way to say it?
I think so. Yeah. I mean, that's there's been hundreds of millions, if not billions of years pushing these organisms in this direction. And they are very well adapted to this environment. In fact, some of the research we're doing in my lab right now is to examine the nutrient concentration envelope of these taxa. And if you provide them with too many nutrients, they definitely give up the ghost. They actually die. It's not that they just slow down. And so their growth rates are very different than a classical monode relationship for E. coli, where as you increase the nutrient concentration, the growth rate goes up. It's actually sort of more of a bell-shaped curve for SAR11, where they go up for a little bit and then they start to come back down. And that's what I would call sort of a true oligotrophic signature of a microbe. And you'd be surprised how few organisms we actually have that kind of data for.
It's a really fascinating idea, and it suggests to me that the conditions where you find it normally are not terrifically changeable because of that fine-tuning. There's an awful word that I use, please forgive me. It's been Darwinoed by evolution. But in a way, I think that that's true. I've heard the term streamlining, the whole 9 yards on that. The idea is this organism has been streamlined biochemically and genetically to these conditions, which would be oligotrophic. Are there other types of conditions for which they're well adapted?
Yeah, well, the open ocean versions of SAR11 are certainly swimming around in an oligotrophic environment. But there's plenty of SAR11s that you find in coastal systems too. And in fact, we've spent some time recently studying a brackish water subclade of SAR11 that's adapted to intermediate salinities. We also are studying a freshwater version of SAR11, which we have as far as we know right now, the only cultured representative, although I'm assuming somebody will get other versions of this soon, which we've named Candidatus Fonzibacter ubiquus, strain LSUCC0530. I think we have a picture of that. So that's a freshwater version of SAR11. And so you find these SAR11s kind of all over the place, and including in places that are not known to be oligotrophic, like coastal environments. So we're in the process of researching exactly those types of things, like what are the nutrient requirements of different strains? What's the envelope of concentrations that they can deal with, and how are they differentially adapted? But it's important to note that the last common ancestor of this group is right now, all the evidence points to it being a genome streamlined, or at least genome reduced, small sort of organism that possibly was already adapted to oligotrophic environments. So there may be sort of a Mueller's ratchet effect happening here, where as these organisms diversify to different environments, they're already sort of stuck with a minimal genome, and their evolutionary processes just continue to winnow away at that. And it doesn't lend itself to to major changes in adaptive capability. So even though they may not mind a higher nutrient concentration under some circumstances, they didn't evolve to be specialized for that. So we're
That's
working on that right now. But yeah, I mean, you don't find them in places that are really nutrient rich, like human guts or sediments or things like this. So that there's only so much of it. We can only push this envelope so far with this not skepticism. We can only push this envelope so far with this speculation.
Well, it's interesting because, a lot of times we look for patterns or we look for hypotheses to explain things, and then we ignore observations that don't fit that because we really like that particular hypothesis that we have. And so what I'm hearing here is you're just looking at what they're doing. Previous episodes of this podcast have talked about how commonly bacteria can evolve even within the laboratory in some conditions. So it's not surprising that that is happening in the largest laboratory, which is our whole ecosystem. And so I think I do want to talk a little bit more about the physiology of SAR11, but the issue of how you isolate these is, to me, very interesting. Would you mind going through that?
Sure. Yeah, we use a technique that was refined by Steve Giovannoni's lab, which they termed high throughput culturing. We tend to refer to it as deletion to extinction culturing. But either way, both of these are sort of ambiguous terms and don't mean a lot just with the names. The idea is that you're going to, instead of using plate-based medium, you're going to use a liquid medium. And part of the reason for that is, as you mentioned, this great plate count anomaly, which was, and then they have their own little individual well to grow in, and they can tootle along at their own growth rate without needing to worry about competition from faster growing copiotrophic type organisms. So this pre-isolation from the dilution is the other side that matters. First, it's the medium, then it's this dilution-based approach. We have sort of taken this to a new level recently, where we're starting to automate this process. We are using 96-well pipetting robots and high throughput identification processes. And we're starting to make this something where we're doing this in sort of thousands of culture attempts in any given experiment, or multiple experiments running simultaneously. And I have some talented grad students in the lab, including Zach Henning, who are really pushing this forward with an automated process. So the technique is still very basic and still follows those same general guidelines, but we're pushing it into an automated version. And I think we have that graphic here that shows some of the basic steps where we're interacting with the robot and the different phases of identifying the cultures and moving them through the process. So that's the, I guess, longish description of how we do the culturing and the isolation.
Oh, no, I think it's very important to hear it that way. We recently had a podcast guest who talked about using fluorescence activated cell sorting to kind of push out a couple of different cells per droplet. This isn't that different. And I love the idea of it, because when we look at those liquids, cloudy or clear, without knowing how many cells are in there, I mean, how do we get down to that kind of level? And so some of this turns into essentially engineering. And this is where we've done some, not we, but science has done some really wonderful stuff with this to be able to actually look at this. And so what started with an idea in immunology to be able to make hybridomas that or make monoclonal antibodies can be used in microbiology in general now. So I think it's a wonderful idea.
Yeah,
your turn.
We're actually, the process that I described to you is what we would call an untargeted approach, because it's not focused on any individual organisms. It's just sort of collecting whatever's out there. And so it tends to be successful for isolating organisms that are more dominant in the community. And that's a good one for SAR11, because they're usually one of the top OTUs in any marine environment that you look at. However, if you're trying to find organisms that are in lower abundance or harder to pull out of a community, these sort of targeted approaches are really, really valuable. And we're actually starting to explore that as well with fluorescence activated cell sorting after using specific tags to identify individual tags of interest. So yeah, that's the future for sure, if you have access to the type of equipment that you need to do that. And Mircea Podar's lab with Carissa Cross had that wonderful paper in Nature Biotechnology a few years ago that showed that you could use antibodies for this purpose. And there's been some other advances. Phil Hugenholtz and Jude Tyson have shown that you can do it with fish probe tagging. And yeah, it's very exciting time for grabbing the organism that you want and separating it away from the community. But the medium that you put it in is still going to be the critical ingredient for success of culturing, because you can do all the cell sorting you want, but if you don't know what it needs to live and you can't predict what its growth conditions are, then you will fail.
So my understanding with these organisms is they grow up to a particular level under these conditions, which is a little bit different than what we're used to with commonly cultivated bacteria. For example, when I showed that flask of photobacterium, that's easily 5 times 10 to the ninth CFUs, cells per mil. But when you grow up SAR11 or Pelagibacter in general, you get 10 to the 7th?
Well, it depends, Mark. We actually have made some breakthroughs recently where we can now culture these to about 10 to the ninth. It involves increasing the carbon concentration, but you have to be very careful about the ratio of the carbon with the nitrogen and how much nitrogen and what type of nitrogen they're getting. There's also an issue with phosphorus as well, or phosphate. So it turns out that these organisms are finicky in terms of the types of these organisms are finicky in terms of the ratios of different compounds that you're providing them with. And so if you just bomb them with high concentrations of stuff, they wind up dying. But if you provide a very prescribed ratio of different compounds, you can actually grow them up to much higher cell densities successfully. So we're just beginning to explore this. Steve Giovannoni postulated this as a major issue for these organisms a long time ago, and it began to explore this when they were first developing artificial media for SAR11. The idea that it's not just about concentration, it's about the relative ratios of compounds in the medium, and that they're tuned to certain aspects of this. And we're doing some experimentation on this, but it wasn't our idea. It came from Giovannoni's lab a few years ago, but we're definitely seeing evidence of that. And we've been able to manipulate it to some extent to take advantage and get these organisms up to higher cell densities than you would ever see in nature. So that's not necessarily environmentally accurate, but it saves us a huge amount of work when we're trying to do proteomics or metabolomics or any of these other advanced techniques, because we can now use typical few 100 mil culture volumes instead of having to grow these things in 20 liters, which is what has been done in the past to get proteomics data for SAR11.
So can I shift the focus a little bit about how the genomes have been streamlined? This organism, one of the most ubiquitous on our planet, what has it maintained and what has it lost? And I know that the latter question is very broad.
Yeah. Yeah. Well, let's start with what it's maintained. I mean, these, like I said, they're obligate aerobic heterotrophs. They're typically capable of oxidizing a variety of different small carbon compounds that we would find in as typical constituents of dissolved organic matter out in the oceans. It depends on the strain. Some of them have the ability to oxidize a variety of C1 compounds, and some of them don't. Some of them have the ability to use some sugars, and some of them have a lower repertoire for that. Some of them can use certain amino acids, and others can make more amino acids than some. So typical central carbon metabolism type processes are pretty well conserved. But some of the weird things that they've lost are, for example, the ability to use oxidized versions of sulfur. So here you have the most abundant microbe in the oceans, And they don't have the ability to use the most common sulfur source, which is sulfate. It's 30 millimolar in marine water. Typically, it's everywhere. It's the third highest ion in seawater, and SAR11 can't use it. Most microbes that swim around the oceans have the ability, well, maybe not most, but a lot of microbes in the ocean have the ability to assimilate sulfate as their sulfur source. SAR11 requires a reduced and, as far as we can tell, organic sulfur source, like something that you would find in amino acids, for example. That was a Nature paper a number of years ago because it was a complete sort of mind-blowing phenomenon that these organisms were so dependent on such a strange sulfur source when they're surrounded by such an abundant compound like sulfate. They've also lost a number of other pathways that are typical in marine microbes. And one of the things that's a hallmark of SAR11 is that they have a far fewer number of sigma factors than a lot of other microbes, even as would be predicted for their genome size. So they're sort of like lower on sigma factors than you would expect just based on the size or the amount of DNA that they have. And what that leads to is that there's a tremendous amount of constitutive expression of genes in SAR11. They really don't have the same regulatory capability as a lot of organisms do. And so they're sort of they're not as flexible. They do have regulation. I don't want this to be misunderstood. And there's a wonderful review on this by Stephen Knoll and Steve Giovannoni recently in bacteriological reviews, microbiology and bacteriological reviews. So that came out just last year or the year before. So if you want to read about the regulatory thing, that's already out there. But that's another weird aspect of SAR11 that lends themselves to some unusual capabilities. So it sort of depends on the strain, but there are definitely some losses. Some of the strains are glycine dependent. This HTCC 1062 is glycine oxotrophic, so you have to supply glycine in the media or it won't grow. But that's not true for all SAR11s. Groups in some of the other subclades can make their own glycine. So it really does depend on which SAR11 you're looking at.
So it's really like nature is continually filing away evolutionarily about things, and we don't necessarily see what the selective advantages are, but they are clearly there. And so, this idea is fascinating to me. And this business of not having as many sigma factors as you might see in a typical heterotroph, it's really interesting because those sigma factors, they're like transcription factors in eukaryotes. And what that means is they help turn genes on and off. Kind of makes sense. I want to say because of the constancy of their environment, but I don't know if I can really say that, as we discussed earlier.
Yeah. Well, the ocean environments, depending on where they live, do change, and you see phytoplankton blooms and things like that. But they are certainly adapted to the sort of normal milieu of dissolved organic matter that might be coming out of any cell that you might see, right? A lot of the compounds that SAR11 likes to eat are the types of things you would expect to be floating around when cells burst open from, say, viral lysis or something like that. So, TCA cycle intermediates and these types of things that are going to be very, very common constituents of dissolved organic matter under normal bacterial warfare and in the oceans.
It's interesting to me because there was a move recently to kind of move away from a Marshall view of microbial interactions, and that everything was much more, I'm not going to use, because when you say symbiosis, that includes negative interactions as well. But you know what I'm getting at, kind of a Margulian view versus a more Marshall view. But I mean, when people give me choices like that and say, which do I prefer? I say yes, because I don't think you can really say that one is more important or more common than the other. I think there's constant jostling up for advantage. And I think that's very clear. That's really interesting.
I mean, the way I teach antibiotics to my microbiology students is that, we have antibiotics for our own purposes to kill microbes because microbes created them for their purposes to kill microbes. So, we're just co-opting a biotechnology that's been evolving for millions of years, billions of years, really. But that's all for killing. So there's not, you can't totally get away from the martial analogies. And certainly when you involve viruses, there's a lot of death and destruction out there at all times, and it is very easy to see the analogy. So I would be hesitant to try to make it seem like it's less of a sort of death dominated scenario. I mean, the turnover
But
rates are estimated to be extraordinary in some cases. Very high percentages of microbes die every single day and are replaced by their non-dying brethren that divide. So it's a lot. It's a lot.
it is. And once again, it's like I kind of, Rudyard Kipling wrote these just so stories from the old colonialist British perspective. How the elephant got its trunk, why a giraffe's neck is long. They sound great to you, but they're not true. And they're not
Yeah.
complete, even if there's a little truth to it. And that's the same thing here. We don't have a microbial perspective.
Right.
And what we
Yeah.
have is a
True.
human perspective. So as we start to kind of wind down from our session today, what would be something that you could say to a person who wasn't a marine microbiologist about the value of the pelagibacters? Do they do anything that impacts our world?
Right. Well, they are heterotrophs, which means they respire organic carbon and they turn it into CO2. This means that they are one half of the carbon cycle in terms of CO2 going into fixed biomass and or fixed carbon compounds and then being put back out into the atmosphere. The other thing is because they are so abundant, they make up a very significant portion of the actual carbon in the oceans themselves, in their biomass. So you have kind of two major things that make them important. It's their ability to sequester carbon as cells and their ability to push CO2 into the surrounding oceans and then therefore through the carbon cycle in the atmosphere. So when ecosystem modelers are trying to understand carbon cycling in the oceans, they have all these components for phototrophs. But what they don't understand and what many models typically leave out is the heterotrophic component. It's usually just an arrow. It's just a flux value for some amount of fixed carbon going back to CO2. And colleagues like Naomi Levine, one of my close colleagues here at USC, are working very hard on trying to break that into much more well-defined quantification and modeling. And so people who are studying abundant heterotrophs like myself are of value because we need to understand how those heterotrophs fill into these carbon cycling arrows, right? We need to understand how the metabolism of these heterotrophs fits into these global ecosystem models to make them more accurate and to make them more and to make their predictions better. So yeah, those are the main reasons why SAR 11 matters. And if they weren't so abundant, maybe it wouldn't be as interesting of a question. But because there are so many cells, it's estimated somewhere around 10 to the 28th cells in the world's oceans, which is orders of magnitude more SAR 11 cells than stars in the known universe. They matter.
I couldn't agree more. And it's a good example of that microbial centric view that we all need to have. I mean, it's like when people tell me that trees make all the oxygen that we breathe, I sit on my hands and I look sad because it's not really true. But then again, I'm known for being a microbial centrist. I've been called that many times. So the last thing that I wanted to discuss with you, and it just for a couple of minutes, is you've created some course-based work around this for undergraduates. And I
Yes.
love that idea, and I'm going to link to some of your papers on it, but do you have some thoughts to share?
Sure. Yeah. Well, I guess this all comes back to the beginning of this story where I told you that I didn't have any lab experience when I finished college, except for the labs that I had taken in school. And part of the reason why I didn't have any lab experience is because I thought labs were boring and lame. We knew it was going to happen. We were using notes from somebody else's lab notebook, and we were copying making the lab notebook out of like 20-page lab reports from some template that we were copying from that was handed to us by the TA, and trying to find out that amylase does what amylase does, and loading tip boxes with pipette tips and doing all kinds of things that I found to be quite boring. And so it wasn't until I became an actual working scientist that I realized how much fun working and doing real science and doing real experiments and making real discoveries actually is. So the CURES, course-based undergraduate research experiences, are a format for introducing undergraduates in their typical introductory biology courses to real research. And this was a strategy that I encountered at LSU by Bill Wischusen, who was running the CURE he was sort of the czar of the CURES at LSU in the undergraduate biology courses when I was a faculty there. And we got involved through that process and started developing a cure for isolating new microbes from seawater based on the methods that I've told you, without all the automation, doing a simpler version that the undergraduates can do. And then we've sort of pursued additional versions of that. We've got a cure where we have students characterizing microbes, so doing physiological experiments and determining their temperature, the range of temperatures they can grow at, or the range of salinities that they can grow at, or pHs. We've also, and those curricula we've published, but we've also started involving students in genome assembly, metagenome assembly. We've got people involved in reconstructing metabolisms and other things like that. And so depending on the course, we've actually kind of branched out a bit. But I find that, one, it's more interesting as a faculty member to involve students in these types of activities. Two, it allows them to generate data that can be useful for real world projects and can wind up getting them on publications. For example, one of our courses a couple of years ago, we assembled a bunch of metagenomes from a microcosm experiment, and all the undergraduate students are co-authors on the genome announcement of the 50 some odd mags that we published from that research. Or it might have even been more. But anyway, that's an example of getting undergrads not only involved in real research, but then giving them products that they can put on their CVs and helping them move their careers along. Not to mention all the training they're getting in state-of-the-art techniques. So it's a win-win-win in all kinds of ways. And I really like this approach.
I do too, and many people use them, and they've been wonderful everywhere for exactly the reasons that we're saying. Many of our viewers or listeners may be used to those kinds of labs where you jump through the hoops. That period has passed. There is no substitute for when a student finds something out that no one had seen before that they found out. And you've just given another great description of exactly that. Cameron, a lot of people think that doing microbiological research, you're at a lab bench all the time, and that can be important, but you have an entire other laboratory to work at. Can you tell us about it?
Right. Well, part of being a marine microbiologist is that we do go to sea from time to time, and that means being on research vessels that travel to a variety of locations. So our hypoxia research, as I mentioned before, is being done, or has been done, in the northern Gulf of Mexico, has been aboard the RV Pelican, and we've done 4 cruises out there in the last few years. This involves sampling by dropping CTDs over the side, which have large bottles attached to them called niskins that can collect water from various depths. Those come up off the side of the ship and we haul them aboard. There's a picture of my grad student, Zach, helping one of the crew members bring a CTD on board. And then we collect water out of the CTDs into small bottles. Shelby Barnes, a technician in my lab, accompanied us on some of these cruises. And we bring that water from these various sample collections into the lab on the ship itself, where we have a whole bunch of equipment set up to do a variety of incubations and other types of experiments while we're underway. And then we repeat that at different locations in different places. We've also had a chance to, as I said, under studying the trichodesmium microbiome, take cruises out of Bermuda with Dr. Eric Webb's lab, who's also a colleague of mine here at USC. And we were lucky enough to have a full rainbow display on one cruise that we captured fortunately, we were able to capture in a photograph.
Cameron, it's been delightful speaking with you today. I have a million ideas to chat with you about in another way, and I'll do that offline. And I know in your copious free time, you'll get back to me at 2 a.m. I understand that. But I really appreciate your time and your effort, and it means a lot to me that you're here, and I'm so happy to have shared some time with you. Thank you so very much.
Thank you, Mark. It's been my pleasure. I'm honored to be on your podcast, and I look forward to seeing all the ones that come next.
Well, thanks again so much. Best wishes.
Cheers.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, as usual with wonderful, wonderful links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me at the Biology Department of the University of Puget Sound in Tacoma, Washington. Dr. Cameron Thrash is in the Department of Biological Sciences at the University of Southern California in my old hometown of Los Angeles, California. Many, many thanks to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope you've enjoyed being part of our Quality Quorum today. See you next time on Matters Microbial.